Our next speaker is Kathleen Caron from the University of North Carolina.
Okay, thank you very much to the organizers for inviting me today. I'm really excited to be talking about a vessel system that we haven't heard about yet, the lymphatic vascular system. This is also very humbling for me to be representing basically an entire field of research. And so when I put this talk together, I weaved in some work from our laboratory, but it won't have that much data because I feel that really the work from our laboratory, I want to use it as an example of what are the pressing questions in the field of lymphatic biology, what are the tools that we're using now, and what are the future things that we really need to be focusing on as a field. The adult lymphatic vascular system is parallel to the blood lymphatic vascular system, and it's a completely separate system. So the blood vasculature and the lymphatic vasculature do not connect, except at one place where the thoracic duct drains all of the interstitial fluid from the body into the subclavian vein. And when you read most medical textbooks, they only devote about 3/4 of a page to a page on the lymphatic vascular system. And what those medical textbooks will tell you is that there are principal functions of the system to drain interstitial fluid, and in doing so, maintaining fluid homeostasis. The lymphatics are very important conduits for the trafficking and the maturation of immune cells as they connect with the tertiary lymphoid organs. And of course, they play a very important role in absorbing lipids within the GI tract. Every villi within the intestine has a lacteal, a lymphatic vessel, and that is where chylomicrons and lipids are absorbed. And so they're quite important in metabolism. We're learning quite a bit about differences between the structure of lymphatic capillaries and blood capillaries. So for example, lymphatic capillaries do not have a basement membrane, and there are no parasites associated with them. The flow of lymphatics is the interstitial pressure from the outside is sensed by these fibrillin-containing anchoring filaments, which changes the permeability between lymphatic endothelial cells. And these barriers and junctions are quite different than they are in the blood vasculature. And this fluid then, through peristaltic-like contractions of lymphatic smooth muscle cells, which are sort of a hybrid muscle cell between a cardiac muscle cell and the traditional vascular smooth muscle cell, causes this peristaltic flow through one-way valves. And all of this fluid then drains and eventually re-enters the venous circulation through the subclavian vein. So it's quite a bit of fluid, so 120 mils per hour, 2 to 3 liters per day. And so if this goes bad, bad things happen. And so deficits in the function of the lymphatic vascular system are associated with lymphedema, and there are several forms. Primary lymphedema or congenital inherited forms of lymphedema are rather rare, but remarkably, we heard earlier about sex differences in the vasculature. It is a well-recognized clinical epidemiological knowledge that women are primarily affected by congenital forms of lymphedema, and sadly they first develop these symptoms right at puberty. And so clearly sex hormones are playing an important role in the regulation of lymphatic vasculature as blood vasculature, but these studies are completely unknown and are not currently being addressed in the field. Secondary lymphedema is very common. You probably know someone, your wife or your spouse or your neighbor may have had this wonderful survival surgery of removing sentinel lymph nodes, which will help save people from breast cancer, but these individuals will suffer the lifelong consequences of lymphedema in the arms. And of course, filarial lymphedema or elephantitis, we saw an image yesterday about this, very prevalent worldwide in underdeveloped countries. But there are a large number of other syndromes and disorders that also have deficits in the lymphatic vascular system. The lymphatics also take the forefront in terms of metastasis of tumor cells, and we spend a lot of money and a lot of energy on anti-angiogenic therapies, but often tend to forget that these tumors are also invested with lymphatic vessels. And in fact, the primary conduit for those tumor cells to the sentinel lymph node is through lymphatic vessels. And surprisingly, despite all of these clinical problems, there are currently no pharmacological treatments to target the lymphatic vascular system. So patients with lymphedema are recommended to wear compression garments or undergo massage therapy. Unfortunately, those have to be rigorously held to on a daily basis so that the edema doesn't lead to inflammation and fibrosis. In the setting of tumors, there have been some successes with anti-VEGF-C therapies. However, recognizing that VEGFs also target the blood vessels, it's not an all or none. And so there are no targeted lymphatic therapies. So for this reason, there is a critical need for identifying new genes and pathways that are specific to lymphatics. And much of that in the past 10 years in this field has come from lessons that we've learned through mouse embryonic development. And so this is an older image of the stepwise process of lymphangiogenesis, where lymphatic precursors our progenitors, I should say, arise from venous endothelial cells early in development due to the transcriptional reprogramming of these cells by SOX18 and PROX1, these lymphatic-specific transcription factors. And that propagates the expression of these cells and the fading of these cells to lymphatics, which bud away from the jugular vein and form the lymph sac that then branches and forms the mature lymphatic network. And in developmental biology, many people in the field will discover that they're embryonic knockout mice. Maybe we could dim the lights so we could see it a little better. Can we do that? Yeah, so one of the common features when you have abnormal lymphatic development is this hydrops fatalis or edema. There's also blood lymphatic mixing if the blood vessels are not appropriately separated from lymphatic vessels, and patterning during development. And so these genes are a short list, and Guillermo Oliver has very nicely expanded on this list. And like many people in the field, our lab came to lymphatics quite by accident, because we were interested for a long time in studying the properties of adrenomedullin, which is an angiogenic factor that's highly enriched in endothelial cells and smooth muscle cells, and clinically relevant in preeclampsia and cardioprotection, that signals through this G protein coupled receptor and accessory protein. And when we knocked out each of these genes, we discovered this phenotype of this hydrops fatalis in each of these animals. And that's associated with a complete block in lymphangiogenesis at mid-gestation. So the lymph sac here in the wild-type animal is about 2/3 the size of the jugular lymphatic, but in these knockouts, there's a developmental arrest. And this is 3D optical projection tomography showing you this absence of these large jugular lymphatics that then causes this lymphedema. So this article, which was nicely a commentary by Mark Kahn at University of Pennsylvania, raised a very interesting conundrum that we struggle with in the field of lymphatics, and that is that most lymphatic factors are also angiogenic, and they're also expressed in the blood vasculature. And so it raises the question of what is the difference between a lymphatic endothelial cell and the neighboring blood vessel, and how do we distinguish how growth factors influence these. Now, in our study, we focused on the fact that the receptor, the G protein coupled receptor for adrenomedullin and RAMP2, are regulated by PROX1, this lymphatic specific transcription factor, and in a way sensitizes the lymphatic endothelium. So that's one example where you could have just differences in levels of these different receptors. But more recently, actually just last week, we've made an additional discovery that the lymphatic endothelium is also enriched for a particular type of chemokine receptor called atypical chemokine receptors, or decoy receptors, as these decoy ducks here on the cover represent. And what we discovered is that absence of this receptor results in precocious and accelerated lymphangiogenesis. You can see here the lymph sac of these knockout animals is very large, because there is excessive adrenomedullin function, because this receptor functions as a decoy trap for adrenomegaline as well as other chemokines and cytokines. And so we can completely reverse this phenotype by genetically crossing it onto haploinsufficient mice. But I think so this is fun for us because we were obsessed with adrenomegaline, but in a broader context for the field of lymphatics, it's important to recognize that there are these specialized families of decoy receptors. There are only 4 of them, and they play really critical roles in ensuring that chemokines and cytokines that are at a low level within the lymphatic endothelium. Because as the immune cells are trafficking through the lymphatic system, you don't want them activated. You don't want abnormal inflammation within the lymphatics. And these decoy receptors represent a paradigm in which we believe that this is happening throughout the body, not just through in embryogenesis. So these types of studies, again, are grounded in developmental biology, and they allow us to explore the functions of these types of growth factors on the lymphatic endothelium. But then what I feel is what happens in the field is that they sort of promote us to begin to ask what sorts of questions we could ask about lymphatic vessels in adult disease. And so, for example, many labs, including ours, will use orthotopic tumor models to dose a tumor and provide a tumor with either no or excessive lymphangiogenic factors, and then look at the growth of those lymphatic vessels in the tumor. And so this is an example from our lab where we have a dosage of adrenomegaline, and that induces lymphangiogenesis within the primary tumor, and those lymphatic vessels are draining, and they're large and they're draining, and so the sentinel lymph node is enlarged in these animals, and it seeds and soils that lymph node for enhanced lymphangiogenesis, which ultimately leads to an increased hematogenous metastasis of tumor cells to the lung. And so in this way, we can develop models where we can actively study lymphangiogenesis in adult animals, and then we can use these models to start to use pharmacological therapies to inhibit this process. More recently, many labs, including ours, are using Krelock's approaches to look at the role of lymphatics in adult animals, and I think this is where the field stands right now, is really trying to explore what are lymphatics doing throughout the body and in different vascular beds. Here we used a global inducible tamoxifen line to delete the receptor in adult mice. The eye is the first thing that's affected, and so there's marked corneal edema due to lymph stasis within the corneoscleral lymphatics. The skin also has deficits in lymphedema, and we see peripheral edema in the limbs. And the intestine is unable to absorb lipid in these animals. So over a long time, these animals fail to thrive, and there is an accumulation of chyle within the lymphatics of these animals. And all of these phenotypes perfectly recapitulate the clinical syndrome of human lymphangdictasia that's seen clinically, and you can see some of these human patients, and all of these features are associated with all of these syndromes. So what have we learned? We've learned a lot from developmental biology. In fact, the entire field, I would say, with a few exceptions in human genetic studies that have identified genes in human patients, most of what we've learned in the past dozen years have come from developmental studies with these mouse models. And that has led, of course, to new genetic tools. And so we're very fortunate in the field now where we have 2 Prox-Cre mice that will enable us to knock out genes specifically in lymphatic, and those were generated by Guillermo Oliver at St. Jude's and Taja McKeenan at University College London. So this is a new opportunity for the field. We know a lot about form and function, and in fact we had to do a lot in the field as a whole. David Zaveda's group has characterized these very specialized lymphatic smooth muscle cells, and Donald McDonald's group at San Francisco has done elegant work characterizing the junctions of these cells that are quite different. They are oak leaf and interdigitating and have these structures called complexus adherens, and so they're very different than the blood vasculature, but quite specialized. Heterogeneity is a term that we've heard a lot in this conference. This is true for lymphatics, not only understanding how lymphatics are different from blood, but how lymphatics are different in different organs. For example, the heart is full of lymphatic vessels, but we have absolutely no idea what these vessels are doing in myocardial infarction, in hypoxia, in heart failure, and these questions are not, to my knowledge, currently being addressed. In the gut, this is optical projected tomography of the tube of the gut here are the submucosal lymphatics, and here are the villi. Look, and this is specific for lymphatic endothelium. So look at how much lymphatics are in the gut. And so clearly, these vessels are incredibly important for metabolism and lipid absorption. And in the eye, we need to rewrite the textbooks. The textbooks tell you that there are no lymphatics in the eye. That's clearly not the case. Elegant studies by Kariali Talo's group in Helsinki and Guo Young Ko's group in South Korea, just published a few months ago, have shown that Schlem's canal, which is critical for draining the aqueous humor that's involved in glaucoma, actually has features of lymphatic vessels, and that was published in JCI, and we were honored to write a commentary article on those exciting findings. So what do we need to do in the future? I think we need to continue to do development because this is where our knowledge base is coming from. But also that knowledge base can come from clinical studies where we have patient cohorts and we can begin to identify genes in the clinic. But there's something that's really limiting clinicians, and that is that the diagnostic criteria for lymphangiogenesis is not well established. And so clinicians need to get together and develop flowcharts and differential diagnosis and try and bin these patients into the different type of diseases and syndromes. And of course, improved imaging will help achieve that. We've talked a little bit about cancer, but I will just touch on how the trafficking of tumor cells as well as immune cells to the tertiary lymph nodes is a really fascinating process. And the way that the lymphatic endothelium presents those cells to the tertiary lymphoid organ is a really fascinating process and very important for adaptive immunity. Inflammation, anytime you have inflammation, you have lymphatics. And of course, within the gut, the role of lipids, reverse cholesterol transport, and the fact that lymph fluid itself is very adipogenic means that the lymphatic vascular system is going to be principally involved in matters of obesity and diabetes. So if you find this interesting, I'd encourage you to please look at the March 2014 issue of the JCI. We at the board were excited to ask Dancho Karajewski from Vienna to be the series editor for this and there's a dozen articles here, and we really challenged them. We told them, don't write the same review you always write. Write one that really, pushes the boundaries and says, what does this field need to do in the 5 to 10 years? And they've done an excellent job. So on behalf of the folks in the lab who contributed to the data I showed, and very humbly on behalf of the lymphatic field, thank you for your attention, and I'd love to take any questions.
We have time for a couple questions.
Hi, real quick. The prevailing teaching in the lung is that the lymphatics stop in the perivascular spaces of vessels about 100 microns in diameter and don't protrude into into the working capillary units. That's been questioned, I guess, with the D240 antibody. There's some pericapillary evidence for lymphatics.
Yep.
My question is, what's your, is that correct? Is that a correct
Yeah.
teaching, and do we know why, or what
Yeah.
the exclusion signals are for why it doesn't?
That's right. So the textbooks were based a lot on our classical markers, which back then, the D240, Live 1, and we didn't have many markers. Now we have more markers, and we understand the heterogeneity of this vascular system. So Schlem's canal is a nice example. It's not an all or none. Not every lymphatic gene must be on to make a cell behave like a lymphatic endothelial cell. So perhaps in the lung, if you were to use an expanded repertoire of markers that are now available because of the mouse studies, you might discover that, in fact, there are lymphatic-like cells within those structures. Yeah.
I'm going to take Jonathan first because he's been trying to get a question in for a long time.
Well, thank you. John Lederer, University of Maryland. Fascinating story. So first question, I was wondering what the distribution of the lymphatics in the heart was compared to, say, the vasculature.
Yeah. So that image that I showed you
Yes.
were the lymphatic vessels on the surface of the heart. Everywhere we see a coronary vessel come down, there's a lymphatic right next to it.
But let's say you go into the heart itself where you
Into the
have
heart
one
itself.
capillary per
Yep.
heart cell.
do
What's the vascular representation?
Yeah, we
like?
do see that. We have the data, and we could quantitate it, though we haven't. Not every capillary junction within a cross-section of the heart will have a lymphatic. It's not, it's less dense than the blood vasculature.
So the related question is filariasis. Does it have any cardiac consequences or consequences for the eye?
Oh, what a fantastic question. I don't know. So most of the filariasis affects the lower extremities, because the parasites, for whatever reason, like to live in the lymphatic capillaries. So it's actually a physical obstruction to lymphatic flow. Very fascinating. But I'm not aware of patients that have that, of having eye complications. They may have heart because of other problems with their circulatory system. Yeah.
One last question over there.
Nice presentation.
Thanks.
Can you speak into the mic?
Of the disease entities that you described, are there clear intestinal lymphatic defects that have been noted, and in particular the lacteals themselves? So,
In all those syndromes?
yeah,
and
those
everything?
syndromes.
Yeah, almost all of those syndromes are associated with protein-losing enteropathy, which is the clinically easy to diagnose. So the extent of the involvement of lymphatics there is not fully characterized, but that's what clinicians need to be working on.
Yeah.
And so Stanley Roxon has a very nice flow chart, but as we learn more, we need to add to that flow chart. I often get emails from patients who say, I've been to my doctor, I have lymphedema, I don't know what to do, they don't know how to diagnose me. Can I take pills of adrenamegalin? And I say, oh heavens no. But clearly the field is in a desperate need of better clinical diagnoses. Yeah.
So we're going to have to move on. Thank you.
Thank you.